Receptors AQA biology. Pinene corpuscle. Well, the pinene corpuscle has specific features, such as they're specific to they're specific to a type of stimuli, which is pressure, and it produces a generated potential by acting as a transductor. And basically, its location is found in the derms of both skin cells, like fingers and external genitalia, and they respond to sudden disturbances and vibrations. So the structure, you have layers of conductive tissue. Yeah, one second, let me get my pointer out. Uh, 1 second. Alright, here's my pointer. So you have layers of conductive tissue with viscous gel between the cell. And then you have your capsule, you have your blood capillary, your neuron, and your nerve ending. And how it works is that, let me get a pen. Wait, this isn't, okay, how can I do this? Alright, so we're going to draw outwards like that. Yes. And how the corpuscle works is you have, if you give me a minute. This isn't, we all know that I can't draw at this point, so. Alright, so you have minus, 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 minus. I'm going to do it in red for you so you can actually see the difference. And then on the outside, I'm going to do green for plus. 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 Alright, so basically this is what the neuron, wait, let me draw vector lines. This is what it looks like within the neuron. And when pressure is applied, almost like you can see in this diagram, pressure is applied, which causes the layer of connective tissue between the viscous layers to be compressed. And as it compresses, wait, let me get a different color. As it causes pressure to compress the layers, so let's see, like that, pressure, we're going to label P, pressure, 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 it actually causes the layers to swap over each other. So these minuses, oops, let's redo that line, that separates them. Causes those minuses, one just, to become positives inside. So it causes the influx of positive ions. And the negative ions go on the outside. Now the internal conditions of a neuron are usually about minus 70 millivolts. I don't know what the thing is called, but I know it's MV would be the written form. So you have minus 70 MV, and due to the influx of the positive ions, it creates plus 40 MV, which is when the generated potential is produced and then it travels along the neuron. So pressure is applied, it causes the influx of positive ions and the out and the release of negative ions out of the neuron, which causes a electrochemical gradient. And then due to this electrochemical gradient, and the positive charge inside the neuron, it causes a generator potential to be produced with impulses traveling along the neuron. Rod cells and cone cells. Rod cells are photochemical receptors and they respond to different wavelengths of light, and the images are seen in black and white. So how do they work? In the retina, rod cells are connected to a singular sensory neuron in the optic nerve. They contain a photosensitive pigment called rhodopsin, and lots of rod cells are converted to bipolar cells, which accumulate. As they accumulate, they create response. Rhodopsin is broken down, and light is detected by the rod cells. Neurotransmitters exceed the threshold value and create a generator potential in the bipolar cell. Impulses travel along the sensory neuron to the brain, and acuity within the rod cells are low as two dots can't be reached and they're seen as one image. So a biology we should have already spoken about resolution is a bit like resolution, which is acuity. I guess you could say in a way you could understand. And around 120 million are found per eye, and they're found predominantly in the periphery region of the eye. So here, let me get my pointer up. Here you can see what I'm talking about. These areas are the periphery, and here you have a rod cell. Now here you have, where's the rod cells? I think I'll draw it actually instead, because I don't really like this diagram. It's good for the cone cells, not so much the rod cells. But you have loads of different, I'm just going to draw lines. I'm going to draw circles because we all know that I can't draw. You're going to draw many, many rod cells. I'm going to draw 4 that are connected to one bipolar cell. That is a horrible, horrible drawing. But yes. And then once the generator potential is obtained, it causes it to pass along the sensory neuron. And that's basically what happens in a rod cell. Whereas cone cells are also a photochemical receptor found in the retina, and it's mainly located in the fovea where there is no rod cells. And the fovea can be found on the diagram in this area right here. Now, rod cells respond to different wavelengths of light, such as which are blue, green, and red at a high frequency, and they have their own separate bipolar cell. So whereas rod cells have many attached to one bipolar cell, it has one rod cone cell attached to one bipolar cell, and they have a photochemical pigment called iodopsin. And how they work is iodopsin is broken down by light, and cone cells trigger neurotransmitters which create a generator potential, which is created in the bipolar cells. The impulses travel along the sensory neuron to the brain, and acuity is very high. So, like I said before, it's a bit like a high resolution, whereas but instead of being like a microscope, it's in your eye. Now, that's all there really is for receptors and rod, cone cells and such. The next video will be about the control of heart rate, and I'll see you in the next video. So if you like this video, then please like and subscribe, and I'll see you on the next one.